All right, it is my pleasure to introduce Wade Naguwa today. Wade completed his Bachelor's of Science in our lab in the Department of Natural Resources and Environmental Management at UH. I actually first met him taking a Hawaiian 101 class at the university. We were supposed to have conversations and ask about each other, and I found out he was a student in NREM wanting to do wildlife work and was able to recruit him to work on our projects. Since then, as an undergraduate student, he worked on poeo, on ungulate research, on water birds, basically everything our lab works on, which was very exciting. And when it came time to finish, he decided to continue on doing a master's project, and it worked out well that he was able to analyze a data set that was being collected by the Division of Forestry and Wildlife on the islands of Maui Nui. So it's really exciting. He's going to be presenting one chapter of his graduate research today, looking at axis deer movement patterns. So with that, Wade, I will turn the time over to you.
Perfect. Um, yeah, well, thank you for the introduction, Melissa. I really appreciate it. Um, Aloha, everyone. I'm Wade Nagua, and mahalo for coming to my presentation today. Before we move on, I'd just like to thank my co-authors for supporting me in getting me to this point, this very long journey. Um, I'd also like to say a quick mahalo to those who have helped with collecting data and helping with the experimental design of this presentation. I would also like to thank those who have helped with the collection and data processing of the game cameras, and the photos that will be used throughout this presentation. So I'm sure all of you are aware that axis deer have been making a lot of headlines in Hawaii recently. And this is due to them just walking straight to the airport, causing a bunch of traffic jams and even showing up on baseball and soccer fields. Yeah, but you likely have never had the full effect of understanding just how big the population of axis deer is on the islands of Maui, Molokai, and Lanai. So, I know it might be a little hard, but out of curiosity, has anyone actually seen an axis deer before, let alone on all three islands, if you can see? Um, Um, well, if you haven't, here's a little video, just kind of showing the actual population of an area on one of the Maui Nui Islands. Yeah. And so, as you see by the video, there's quite a lot of deer in Maui Nui. And just a little introduction about axis deer and their biology. So they're largely grass grazers. They are polygynous. Uh, the reproduction age starts with females in the first year and with males in the second year. About 95% of the females are breeding, and about 27% of males are breeding. So the max age of reproduction is 10 years, and their maximum lifespan is also about 10 years. So they'll be reproducing up until they eventually die. So where are axis deer from? Uh, their native range consists of Sri Lanka, the Indian subcontinent, and Nepal, which Nepal in the range below 1,000 meters in elevation. So as you can see from this map, in the green area, the dots, that's actually their native range. But when you look at the other colored dots, you can kind of see how much they're spread and they were introduced throughout the world. And this is mainly due to hunting and food resources. So, axis deer are one of the many introduced species in the Hawaiian Islands. They were introduced on Molokai in 1868, on Lanai in 1920, and on Maui around 1959 1960. And in the present day, we see a lot of impacts caused by the axis deer. And some reasons why they are so prevalent in Hawaii are due to rapid population growth, the lack of natural predators, and the lack of competition for resources. So, some examples of impacts involving axis deer in the Hawaiian Islands are prolific grazing and habitat degradation. So besides causing traffic jams and distractions at the airport, access deer have a lot of both desirable and undesirable impacts on our islands. So why should we care about access deer? Well, access deer are valued for both recreational hunting and food. In fact, many families on Molokai traditionally have venison on the Christmas table along with other holiday dishes. And also because they're actually good at grazing, which could potentially reduce wildlife or wildfire risk. Excuse me. But when we're looking at this chart, I really want all of you to mostly focus on the yellow lines. So here we can see the different factors that unmanaged feral ungulates have impacts on. So axis deer cause a lot of car strikes each year and can spread diseases. They damage crops and reduce forage available for the livestock. And I know we said that they can reduce wildfire risk. However, if overgrazing does occur, then erosion and sedimentation can damage our coral reefs and fisheries. In a recent study in our lab that I got to help out with, we found that 66% of ranches had losses of forage from wild ungulates when cattle and livestock were fenced out to control for the competitive grazing. Another recent study in our lab showed that ranchers are spending a lot of money on actions to protect the protection, like fenced installation and repairing damaged property. The total extrapolated annual cost in Hawaii come to around 9.2 to $13.1 million dollars. And that's enough to put a lot of small ranchers out of business. It's no surprise that ranchlands and agricultural lands are experience a lot of impacts from axis deer. Their ecological niche is in the central saddle of Maui, which is dominated by ranchlands, golf courses, and private land. The study by Risch et al., which used game cameras, showed where deer are most abundant. But how do axis deer decide where and when to move? If we added ungulate fences, would that restrict movement and how much? Most importantly, what actually drives the animal's movement? So how can we measure this behavior? Typically, we collect a series of GPS points. From this, we can look at the distance they move between points. Um, we can also look at where animals spend most of their time. So this would be home range, which shows us places that are perhaps important to the animal. Maybe there's food or a safe place to rest. If the resources an animal needs are all in small space, they will have a small home range. If they have to travel a long distance to get what they are looking for, then their home range will probably be much larger. And what we can also look at is step length. How is step length used? Basically, how much do they move in a given period of time, whether it be hours, minutes, days, or even years. So going into home range two, what do we know about the axis deer's home range in their native habitats? So recent studies have been shown that males will have a home range of about 3.01 kilometers squared, and females having around 2.02 kilometers squared. And in a more somewhat recent study, they found that males would have around 1.83 kilometers squared, and females having 1.35 kilometers squared for their range. So I guess we can talk about a little bit more on what motivates animals to move in general. Animals will have motivations for why they move, such as food, habitat, reproduction, predators, and seasons. We can test which of these factors is driving behaviors, such as movement or home range, and ask questions like, do these differ in novel environments or compared to their native range? So in this study, we asked two questions. The first question was, does the home range size of axis deer in Hawaii differ from their native range? My null hypothesis was the home range size of axis deer on the islands of Maui Nui does not differ from the native range. My alternative hypothesis was home range size of axis deer is larger on the islands of Maui Nui compared to their native range. And my second research question, which environmental factors are correlated with axis deer movement? My null hypothesis being axis deer movements are all random, and my alternative hypothesis being axis deer movement is correlated with vegetation, temperature, land cover, and sex. So, my study took place on the islands of Maui Nui, which are the only islands where axis deer occur in the Hawaiian Islands. So, for my methods, the first thing we have to do was capture and put GPS collars on the axis deer. So, the capture and release, GPS collaring, and data collection were carried out by DOFA using a three-person team. The callers automatically uploaded data to satellites, and then we were able to download into softwares made by the manufacturers, and we were then able to download the data from that company. And lastly, we quality checked the data and we ran our analysis. So first, for our analysis, I checked the quality of the GPS fixes. This also because some callers were programmed to collect points every hour, every 2 hours, and so on. And I needed to reduce this to match the other callers that collected points every 4 hours. So we chose 4 hours because it was a time interval that we could standardize to without having to sacrifice a significant amount of data. I then overlaid environmental predictors with the GPS data locations so I could associate each point taken from the axis deer with the environmental variables on that day and at that time. So, for example, some of our drivers were vegetation cover, land cover, temperature, and seasonality. Finally, I estimated home range for each deer using an auto-correlated kernel density estimates approach. So just going into autocorrelated kernel density estimates, it is a method for estimating home range that recognize animals move continuously, so nearby GPS points are related to each other. So it uses this movement behavior to produce more realistic estimates of species. Traditional methods treat every GPS point like it's unrelated, but in reality, if a deer is in a certain area at noon, it didn't randomly just jump miles away at 4 p.m. Um, it moved there. So autocorrelated kernel density estimates accounts for that movement. And after estimating home range, I wanted to also analyze the predictors of step length. So when I talk about step length, you can kind of imagine that if you look on the screen, there's 2 points. Step length was calculated as the distance between consecutive points recorded from each deer. Essentially, as a straight line distance, how far did an axis deer move in 4 hours? Keep in mind this is a minimum distance moved since they may have actually traveled a longer distance. So on the left side, you can kind of see what the program was able to give us. At the top, you can see the amount of points that were already taken and the distance from the last point. And then lastly, we ran multiple regression models to determine which factors best predicted step length. So going into our results, the GPS collaring, or sorry, we had 20 total deer that we have collars on 3 different islands. And the GPS collaring started around on May 5th, 2023. And it's actually still continuing to collect data to this day. We have males and females collared for each of the 3 islands. As of right now, we have 10 males and 10 female collars. In total, for our home range, we had around 61,000 total GPS points. And after we standardized it, we had about 41,600 for our standardized points. So just a demonstration of what we're looking at when we're looking at the autocorrelated kernel density estimates and looking for home range. Um, right here is an individual caller. And right now, the home range associated with this specific deer was around 9 kilometers squared. If you look at the map, everything inside the highlighted areas is considered the 95% kernel density estimate, which basically means that's their primary source of or primary area that they stay in. Another example here, we have a female on Lanai, sorry. Um, and this female had about a 5.2 kilometer square range. And lastly, we have another female on Molokai that had about a 42 kilometer square range. So it was a much bigger home range than the other two that we have seen. So in the end, home range size did not differ significantly between males and females. The results were consistent when evaluated using a t-test on log transformed home range. Um, Home range data was not normal, so I also used a Wilcoxon test as the primary analysis. Um, and again, I log transformed the data and confirmed results using a t-test, which produced consistent conclusions. Next, so this is more of the step length side. So what are we seeing when we look at step length in general? Well, first off, as you can see, females are moving much farther than males. And next one, so when we're looking at NDVI or vegetation greenness, some of these females are moving much farther in less green areas. Uh, if you look at the NDVI times 6, our P value is not significant. So this basically means that males and females respond to NDVI or vegetation greenness in a very similar way. When we look at temperature, we can see that deer move less when temperature increases, right? So with temperature, there seems to be an optimal zone. Um, Most of these points were clumped within the 20 to 35 degrees Celsius range. And again, females are moving much more than males. And then we can look at seasonality, so seasonal fluctuations of deer movement. Uh, when we're looking at the year-round season changes, we again see females uh, moving much farther than males throughout the year. And lastly, so we saw that they are most frequently observed in these land covers that are circled on the screen. And they're moving similarly against each other, with an exception to females moving much more in alien dry shrublands. Um, in the green circle, yeah, we are seeing the most counts in the non-native land covers. And so, however, that doesn't mean they aren't in native land covers. So we can also see on the top that they are in native dry shrublands. And just comparing our home range to past studies. So in Sri Lanka, they had around 1.83 kilometers squared. In a study done in Texas, it was about 6.5 kilometers squared for males. And what we're seeing in Hawaii is the home range is actually around 38 kilometers squared, which is much larger. Um, and this can also be said for females with 1.4 kilometers squared in Sri Lanka, around 6.5 kilometers squared still in Texas. And in Hawaii for females, the range is about 11.8 kilometers squared. So to answer the first research question, being, is there a difference between native and non-native home ranges? So yes, we see that there is a difference in home range. These differences may be due to the lack of predators, lower food availability, and or fewer barriers to block movement. To answer the second question, being which environmental factors are correlated with axis deer movement, uh, we see that there are multiple environmental factors that are driving movement. So these movements include vegetation greenness, land cover type, temperature, and seasonality. And so with these results, what can we do with them? Um, we can try to manage by land cover and target areas used more by these axis deers. Maybe we have better predictions for why they move in certain ways so we can then account for it. Um, and if we wanted to look at population control, then these non-native land covers that we saw having a lot of high counts in would be something we would want to focus on the most. However, if we wanted to also limit impact on high conservation areas, such as the native dry shrublands that we saw, we could also put more effort into those areas as well. So management implications for hunting, we haven't, or we still need to talk with hunters about a lot of these management implications. If there are any hunters here that may have some input on hunting, I'd be all ears. I'd be very eager to learn your side and what some thoughts that you have behind this topic. And management for agriculture. So fencing is critical to minimizing impacts to agriculture, and movement within fences will be, driven by food availability. Yeah, again, if we wanted to also limit impact in these high conservation areas, we are at least informed in what areas we are finding them in, and more strategic plans to remove them from these areas are possible. And yeah, lastly, so this was just one of the chapters that I've been working on for my thesis. Um, I believe this is important, but what I invite those in the audience to kind of think about is what is our research gap that we don't know, and how can we address it? In areas that we can't get populations to zero, what should population numbers look like, and how do we get there? So that kind of leads into my second chapter, which is a population viability analysis to test which combinations of removal methods might achieve management goals. Um, and in areas that we can't get to absolute zero, what should the population size um, look like? Yeah, just something to think about. Mahalo. Thank you for taking time out of your day for listening to my presentation.